It's a very great pleasure to introduce Professor Jana Heinerstorfer as today's speaker. I think I should say how we met and why I invited her. We met serendipitously. It was about 3 years ago. I was helping a startup with a really thorny biophotonics challenge. And I called one of the best electrical engineers I know. He's a former TA from 275. He'd gone on to Carnegie Mellon. I said, we're having really big issues extracting tiny fetal pulse oximetry signals from inside the maternal abdomen. He just made one suggestion. He said, get on a plane, come down to Pittsburgh, meet my advisor. So that's what I did. I flew down, I met Jana, I met some of the students in her lab. She drew me beautiful photon bananas on the whiteboard. And today she's actually, with NIH SBIR support, helping the startup, and they're sponsoring research in her lab. And she's very much a hands-on experimentalist. We've done animal studies together. I think my enduring memory of doing an animal study with Jana is she pretty much told me to sit in the corner and don't mess up data collection. That's how she is, but that was a good day regardless. Jana's from Vienna. She had gets her doctorate from the University of Vienna. It was completed at the NIH. She followed this up with a postdoc at Tufts, and then over the last 5 years has built a really diverse group of talented, I think warm-hearted students at CMU. They're working on a really wonderful array of challenges, from non-invasive light-based diagnosis and treatment. As you know, what makes our group cool and excellent is they can go from the theory to building extremely sensitive hardware, testing it in the lab, and then on to in vivo studies. And just 3, I think, are the projects that I know about, and I know there's more. Today's topic, I believe, correct me if I'm wrong, Jana, but will be the cranial perfusion and hemodynamics work that you're doing in monkeys. It has application to strokes. Another project that's gotten really beautiful press is using near-infrared light to identify and monitor lesions in the breasts. And then one project where all of her grad students want to go with her, that's sponsored by the Office of Naval Research, is measuring the perfusion in dolphins and sea lions. And you might want to say a few words on why you're doing that. I give you 3 fun facts from her students. We know who the guilty student is. She's pretty much completely non-functional without coffee. If you do not label the axes on your plots, Jana will crucify you. And on a good day, sometimes her most helpful answer is ja, nein, and you just kind of have to deal with it. So, Jana, the floor is yours. We're standing by for an illuminating presentation.
Thanks, Nevin. I'm going to correct you. The third one is jein. It's a German word that everyone should know. It's really the literal combination of yes and no, which in English would be yo. Doesn't really work that way. But it's the perfect answer when you don't want to say yes or no, and the answer is somewhere in between. And of course I'm sharing the wrong screen. Thank you, Nevin. It is hilarious to me that looking through the attendees that apparently the said student from electrical engineering, who's now my PhD student, apparently advertised this talk also to my lab. So I feel like half of the students on this Zoom meeting are from my own lab at Carnegie Mellon, so they know very well what I'm talking about. But okay, so I am an associate professor in biomedical engineering at Carnegie Mellon, and I'm affiliated with the Neuroscience Institute, as well as electrical and computer engineering and McGowan regenerative medicine, and I think a couple of others, but primarily biomedical engineering and neuroscience. And really, the talk is mostly going to be focused on brain imaging with light, and how do we do that. But it's really in a nutshell, everything we do in the lab is we shoot lasers on people and trying to say something about the tissue, and hopefully at the bedside. So everything boils down to this figure, which is absorption coefficients of hemoglobin. Ultimately, when you shine light onto tissue, there are really 2 things that can happen. You take a laser pointer, you put it through your finger, and you'll see the finger glow. It will glow red mostly, or well, if you take a flashlight, put it in your mouth, your entire head lights up red. And so there are two things that are happening. Light is being absorbed and light is being scattered. And we take advantage of both of them, both of those optical properties. And you can derive equations to describe diffusion of photons, and it's in a diffusion equation and such. But really, in the end, what we do is we take at least two colors of light. And because hemoglobin inside tissue is absorbing light differently depending on if it's oxygenated or not, so that's shown here, the blue and the red curve, we can differentiate between oxygenated and deoxygenated hemoglobin. So any relative combination or relative contribution at each wavelength will tell us something about how much is hemoglobin oxygenated. And what I'm describing to you has been around for decades. That's the very basics of pulse oximetry. And in Nevin, I will not be talking about fetal pulse oximetry today, even though we're doing that. But really, ultimately, what it all boils down to is spectroscopy in its most basic form, two colors of light, and you measure intensity fluctuations that somehow where light has traveled through the tissue. And that gives you a lot of metabolic information of the tissue, specifically about oxy and deoxyhemoglobin. And so for brain monitoring, people have developed instruments. We build our own instruments, and I'll talk about that also. But the basic idea is you take some light source, deliver it typically through optical fibers or LED directly on the head, and the light goes through the skin, near infrared light specifically, goes through the skin, through the bone, interacts with the brain, and due to the scattering nature of photons, comes back out again. Quantitatively, we can describe that, as I said, with a diffusion equation and the red blob on top of this MRI scan here. So it's called the probability density function. So the red blob is pretty much what we're sensitive to when we shine a laser onto tissue and then measure the reflectance coming back out again. That red blob itself is already telling you it's a pretty low resolution imaging method. But what we can do is really build things relatively small and portable. And so there are companies like Artinas that are building wearable sensors where everything is, the battery is all in a backpack and people can walk around with those optical fibers on the head. So the instruments that we use is, I mean, doesn't really matter too much, but typically we have a sampling frequency of those instruments somewhere in the tens of hertz. The typical wavelengths we use are 690, 830 nanometers, so in the near infrared range. Again, two wavelengths at least to differentiate between oxy and deoxyhemoglobin. And then you can solve the diffusion equation, and ultimately this equation down here, which is the simplest form that we're using, which is called the modified Beer-Lambert law, is ultimately a solution to the diffusion equation. And what that says is that if we measure intensity changes, so that's the delta I of each wavelength, we can say something about changes in the absorption coefficient, which then, since we know the extinction coefficient of hemoglobin, we convert into changes of oxy and deoxyhemoglobin. There's a scaling factor in between. That's where the math gets more complicated. That's optical, that's light scattering, and the differential path length factor. But in its basic form, you measure intensity changes on the surface of the tissue. You translate them into absorption coefficient, and that gives you changes of oxy and deoxyhemoglobin. So with that, we can say a lot about neuronal function, or we can say a lot about brain function. And a lot of times we go back and use the hemodynamic signals that I'll be talking about to say something about neuronal function by means of what is called neurovascular coupling. So this idea of neurovascular coupling really just says that when the neurons are active, you guys are listening to me, auditory cortex, as the neurons are firing, they need oxygen and will recruit oxygen through the vasculature. So you can find locally an increase in hemodynamics. And so mathematically that's described by saying if we have some stimuli, some neuronal activation, There's a delayed form of then the hemodynamic signals. So mathematically, that's a convolution product with a response function, the hemodynamic response function. But it really then says we can measure those hemodynamic signals and say something about where and when neuronal activation occurred. And so that's really the basis for this whole field of functional near-infrared spectroscopy, so using near-infrared light. But it's also really what I'm describing here is also commonly used and is always used for functional magnetic resonance imaging, where you measure hemodynamic signals to say something about brain function. But to give you an idea of what that looks like, of course, the best case scenario, I should really eventually get a new image of this. This was more or less the best subject we ever had. But you place an optical sensor, so that's an LED, a light source, and a detector, photodiode on the head. And as a function of time, you measure intensity fluctuations of photons that have traveled inside the tissue and come back out again. In this particular case, I've asked the subjects to perform what's called a finger tapping task. So they tap their fingers, motor cortex is active, again, localized changes in blood flow and hemodynamic recruitment. And so every time the subject is tapping their fingers, you see a change in the intensity signal, so the two different wavelengths. And based on the equation I just gave you, we can convert that into oxy and deoxyhemoglobin. We can do an average, and then you get something that is very specific to functional activation, which is pretty much the hemodynamic response to neuronal function, where you see an increase in the amount of oxyhemoglobin and a decrease in deoxy, and that translates to neuronal functions as measured, for instance, with EEG. So this whole field of functional near-infrared spectroscopy has been around for quite some time. People are using those types of signals to measure functional activation in children, in patients that cannot be put in an MRI machine and such. But really, my lab is centered around this idea of measuring hemodynamic signals, mostly in the brain, but not exclusively. But there's so many more things one can do than look at functional activation per se, which we're also doing. But one of the projects in the lab that we're doing is trying to say, well, can we do all of this non-contact? Do we have to place the optical sources on the surface of the head or on the tissue? Can we do that remotely or well, from a distance? That's one project. Another one is I said that the resolution that we typically get is in the order of this red blob. One of my students is focusing on multimodal imaging and trying to improve spatial and temporal resolution with those methods. So he has shown that if we combine EEG, so instrument that measures neuronal activation directly, combined with a really high density optical system, we can actually do 3D reconstruction of neuronal sources quite accurately. And as I said, I don't exclusively work on the brain. As Nevin said, we do try to develop in collaboration with the company. We're developing instruments for fetal pulse oximetry, but we also look at breast cancer monitoring. And so the idea is that when there's a breast cancer lesion or most cancers have an increase in blood vessel density around it. So hemoglobin, again, is a good biomarker to be tracked, in this case, to be tracking neoadjuvant chemotherapy. And so one of my students, Connie, has developed a handheld device that pretty much, the general idea is add on some optics to a smartphone and image hemodynamic changes over time. And yes, Nevin also mentioned the dolphins. We do have an ongoing project that is trying to evaluate whether or not this near-infrared light goes deep enough before it's being absorbed, such that we can measure dolphin hemodynamics, cerebral hemodynamics as well. So the challenge with the dolphins, very similar to actually fetal pulse oximetry, is that there are large layers of other tissue on top of what you actually want to measure. So it's an optimization problem of how do you choose wavelengths and geometry to be sensitive that deep. It comes with the perks of actually being able to go to Spain and do measurements on dolphins. So that's pretty cool. But really the main project I want to talk about today is how do we use blood flow and hemodynamic signals to look at intracranial pressure, and how do we develop biomarkers to measure, how do we come up with biomarkers to predict patient outcome, especially in the realm of traumatic brain injury and hydrocephalus. And so in terms of intracranial pressure, I want to start out with just the cartoon version of a head, which is a skull. Inside that skull is brain tissue, cerebrospinal fluid, and blood. If any of the three compartments is increasing in volume, the pressure is going up. And so that's happening in hydrocephalus. So that's a buildup of fluid, cerebrospinal fluid, as well as in traumatic brain injury due to bleeding. And both cases, or in any case, if there's prolonged increases in intracranial pressure, that is associated with worse neurological outcomes in patients. And so what is also known is that the brain is actually remarkable in the sense that pressure changes, such as changes in arterial blood pressure, changes in intracranial pressure as well, can be compensated by the brain, which is, the mechanism is called cerebral autoregulation. And so this idea has been around for decades, where the main idea is that cerebral perfusion pressure, which is really the pressure difference between blood pressure and intracranial pressure, cerebral perfusion pressure is driving cerebral blood flow. So within a particular range of perfusion pressure, cerebral blood flow overall is maintained. So that means that the arterioles can actively dilate and constrict such that blood flow is maintained. Outside of that range, you either get not enough blood flow or too much blood flow. In either case, not good for the brain and not good for the neurons. From a clinical perspective, what people have found is that if you treat patients in a traumatic brain injured patients, mostly such that perfusion pressure is within that window where autoregulation is intact, which a lot of times clinicians quantify with a parameter that is called pressure reactivity index. Doesn't really matter for this talk here, but really if we optimize perfusion pressure such that autoregulation is maintained, neurologically, what people have found is that patients have a better outcome. So there's definitely a link between neurological outcome and optimizing pressure inside the head. So really what we're asking then, and there are the two sub-projects I briefly want to talk about, is saying how is autoregulation linked to neurovascular coupling? And the other way around, can we use neurovascular coupling to say something about cerebral autoregulation? Now, what I just said about autoregulation is that there's an optimum perfusion pressure that you would want to target when treating patients, such that blood flow is maintained. And the trouble with this is that while the theory is nice, practically you don't know what pressure is inside the head, except for in very severe cases where clinicians find it necessary to actually drill a hole in the skull and put a pressure probe inside the brain. So it's a very invasive method nowadays. That's the clinical standard. And so my lab has developed a non-invasive alternative to help identify patients that have increases in pressure to then optimize treatment. So I'm going to be talking about the non-invasive intracranial pressure sensing, but then also in terms of neurovascular coupling, I'm going to be talking about how neurovascular coupling influences autoregulation, or what the link is between the two, and that we can non-invasively measure with electroencephalography, as well as the optical method that I just talked about, which is near-infrared spectroscopy. So especially for neurovascular coupling then, what we are asking is, well, we can measure neuronal function with EEG, we can measure the hemodynamics with NIRS, near-infrared spectroscopy. We know that there's a convolution product in between, which is the hemodynamic response function. To give you an idea, the signals kind of look like this. That's a typical visually evoked potential measured with EEG on a visual cortex. The hemodynamics I just talked about in terms of oxy and deoxyhemoglobin. And that's how the systems look like typically. We combine the two instruments as well. And the question we're asking is, does this coupling, does the response function change with perfusion pressure? So therefore with cerebral autoregulation. So a different way of showing this is that if autoregulation is intact, we know this cartoon should hold. If autoregulation is impaired, what does it do to the hemodynamic response function? So what I'm going to be talking about is then non-invasive pressure sensing, as well as then changes of neurovascular coupling with intracranial pressure and perfusion pressure. So for those, the first part for the non-invasive ICP intracranial pressure sensing, we use the optical instruments I just talked about, near-infrared spectroscopy, which is really we just measure intensity fluctuations from which we can derive oxy and deoxyhemoglobin changes. But for the non-invasive ICP sensing, we use a derivative of NIRS, which is called diffuse correlation spectroscopy, which is built by Seth, MIT graduate, who names his instruments, I believe, Charles, because he really enjoyed Boston, so Charles River. But anyway, so he built that instrument. Diffuse correlation spectroscopy is very similar to NIRS in the sense that we use a laser to illuminate the tissue, and we measure photons that have traveled deep inside the tissue, so multiple scattered photons. Some of it comes back out again. But in this particular case, we're using a long coherence laser. And instead of just looking at intensity fluctuations, what we're looking at is the intensity autocorrelation function of that laser, which is plotted here, the G2 curves. The idea is that the decorrelation does go hand in hand with moving particles. So ultimately, the faster particles are moving, the faster the autocorrelation function will decay. And so that's shown here. So what we measure is with the software correlation approach, the temporal autocorrelation function, and the orange curve is shown for fast moving particles and the blue curve for slow moving particles. And the moving particles in particular that we are interested in here and that are important in tissue is essentially blood flow in the microvasculature. So instead of measuring hemoglobin concentration, what we can measure is blood flow changes in the microvasculature, again, in the brain. So ultimately the output is CBF, so changes in cerebral blood flow. The animal model that we have developed is based on a non-human primate model. It's an acute study. Animals are anesthetized. And we use diffuse correlation spectroscopy, so blood flow that can, for measuring autoregulation, and to develop the ICP sensor, we place a sensor, optical sensor as well on the head with NIRS, so that gives us functional activation. And we put a bunch of EEG electrodes, so that's for neuronal function. The animals have a pressure probe, so an intracranial pressure probe in the parenchyma. So that gives us the ground truth of what the pressure is inside the head. And we can modulate pressure in the head by putting more or less fluid inside the head. So that's done through a catheter where we put fluid into the ventricle. So that catheter is attached to a saline reservoir. And so by just lifting that bucket of fluid, by means of gravity, we put more fluid inside the head, and that very controllably changes intracranial pressure. And so over the course of multiple hours, we typically step up the baseline intracranial pressure. And so to give you an idea of what then the data looks like or what we collect, so we collect intracranial pressure from the parenchyma as a function of multiple hours over time. We have an A line, so that measures arterial blood pressure as a function of time. So as you can see, blood pressure does fluctuate, but it does not go hand in hand with intracranial pressure. And then since I talked about perfusion pressure, so that's the difference between blue and red, so red minus blue gives you the green curve here, so that's cerebral perfusion pressure. Optically then, we measure changes in oxyhemoglobin, deoxyhemoglobin, and blood flow. And in this particular case, we oscillated intracranial pressure. Just to give you an idea, then whenever we oscillate pressure, that yields changes in the vasculature. So the vessels are then corresponding or responding to pressure changes in the head, and so is blood flow. So you do see those oscillations. So as I said, the first step, or the first thing I want to talk about is saying that we wanted to develop a non-invasive alternative for measuring pressure in the head. What is known clinically is that the intracranial pressure, when measured invasively, has a lot of features in the data. Every time your heart is pumping, is beating, you will see that in what's called a cardiac waveform of pressure in the head. So the ICP waveform, as shown here, for the cardiac waveform, has a whole lot of meaning to it. So ultimately, when intracranial pressure is normal and healthy, the relationship between the percussion wave and the necrotic wave, or any of those waves, is very well characterized. And when intracranial pressure goes up, the first peak in relationship to the second, so the percussion wave, is very much reduced. So there are a lot of features in that cardiac waveform that go hand in hand with baseline intracranial pressure. And so our hypothesis was that, well, shouldn't that be also true for the microvascular blood flow that we can measure with diffuse correlation spectroscopy? And to give you an idea, so Jason built the DCS, diffuse correlation spectroscopy system, that can measure cerebral blood flow at about 100 Hertz. And showing you this particular example here of a healthy human volunteer, where we placed a sensor on the forehead, that gives you an idea of how the signals kind of look like. So this is cerebral blood flow as a function of time. And every time the heart is beating, you see the cardiac waveform very reliably. And so as a first step, what we did was we performed human subject measurements. Specifically, what the students have done is put a blood flow optical sensor on the head, one on the arm, a bunch of EKG leads to really trigger when, or to know exactly when the heart is beating. And they performed head of bed tilting experiments. So the idea here is that when subjects lay flat on their back versus when they're tilted down by 40 degrees, the difference between those two states is that by means of gravity, we've increased the pressure in the head but in terms of perfusion in the arm, nothing should change. So ultimately saying that by tilting, even though it's just for a short duration for 5 minutes, tilting down those subjects by 40 degrees should increase intracranial pressure, but nothing should happen in the arm. So then again, put the subjects back flat for another 5 minutes. And so what the data kind of looks like is shown here. So that's an averaged blood flow cardiac waveform on the left-hand side for the brain, on the right-hand side for the arm. The red curve is for when the subject is laying down flat. And so what we're expecting is that when we tilt the subject, we should see changes in the left curve, but nothing in the right. Because that's the arm. The arm does not show changes in pressure by tilting. That's exactly what we found. Remember, I talked about the first peak being indicative of pressure in the head, so that's the percussion wave. Indeed, we do find a reduction in blood flow in the cardiac waveform compared to 0 degree, and then tilting them backwards, everything goes back to normal. And the arm does not change. So this data set is really a good indication of saying that the cardiac waveform in blood flow should tell us something about intracranial pressure. And Jason is using those curves in a much more advanced step now, and we have some indication that we can actually quantify impedance of the blood vessels with those type of curves as well. But for the purpose of this talk, let me just talk about intracranial pressure. So again, the intracranial pressure waveform at the cardiac waveform at low pressure should look like this. At high pressure should look like this. And so we're expecting that the cerebral blood flow cardiac waveforms have similar features to that. And so indeed, the type of measurements that we have then done in the monkeys is shown here. So the blue curve is the curves are color-coded with baseline intracranial pressure. The blue curve is for an animal at low intracranial pressure, so healthy, about 5 to 10 mmHg. The blue curve here is the intracranial pressure waveform. Orange is high intracranial pressure. Blood, so is color-coded with high intracranial pressure, in this case the blood flow, versus intracranial pressure. And so the two waveforms definitely do not look the same. I mean, one time we were talking about blood flow versus pressure, so the features should not be exactly the same. But it's a nice example to say that, remember what I said about the first peak relative to the second one? So at low pressure, the first peak is taller than the second. At high pressure, that is no longer the case. So now I'm talking a lot about features of a cardiac waveform. So in the end, what we ended up doing is do exactly that. We do feature extraction. And so specifically, what we said was we have intracranial pulses, cardiac pulses, as well as blood flow pulses. And so we used the cardiac waveform for those animals, averaged over about 60 pulses to improve the signal-to-noise ratio. And then for each of those average pulses, we extract a bunch of features. We take the delay time to the peaks, the height of the peaks, full width F max, area under the curve, as well as mean arterial blood pressure. And we use all of those features combined compared to then the ground truth intracranial pressure. And what Alex, now graduated student, has done is use regression forests to train the algorithm to come up with features that are most descriptive for intracranial pressure. So as I said, we're using all of those features then with the regression forest. And then based on training on about 80% of the data and testing of on 20% of the remaining data and using 5 animals for this work, what we have then come up with is an algorithm to do then the conversion of blood flow, cardiac blood flow, cardiac waveforms in the blood flow change to come up with an intracranial pressure value. So what you should be seeing here is then, or what to expect is that in this figure is that when everything is perfect, those two lines should be a straight line, or that those two values should be corresponding to each other. So on the x-axis, it's the invasively measured intracranial pressure from 5 different animals. On the y-axis is our non-invasive estimate. And in fact, when I plot this all combined for multiple hours of data, each dot here is an average over, I think, 60 pulses over then 5 animals. And what you see is that we find a very good correlation between invasive intracranial pressure. Again, that's drilling a hole through the skull, putting a pressure probe inside the head, versus our non-invasive estimate, so an R-square of about 0.9. A different way of plotting this is the difference between is a blind Altman plot that pretty much shows that we have about a 4 millimeter mercury accuracy based on those animal data sets. So that's super exciting because it's a very quantitative method to measure intracranial pressure. What we're doing right now is, you can also plot that as a function of time and get those really nice time traces of intracranial pressure. What we're doing right now is translating all of this into the clinic, and so we have an ongoing collaboration with the Children's Hospital in Pennsylvania, in Pittsburgh. So that's then a typical hospital card to give you an idea. That's the DCS probe for blood flow without the fibers attached. We have a combined NIRS DCS probe here. We monitor patients in the pediatric ICU, either hydrocephalus or traumatic brain injured patients. We monitor them over multiple hours per day. Every time it says here ICP check, what that corresponds to is that all of those patients have an external ventricular drain to reduce fluid in the head. And every time they're clamping that drain, we can actually measure intracranial pressure, and that's when we can measure optically and correlate our optical measurements to the device. So we have about 14 patients right now, and we're in the midst of analyzing the data. And I'm going to be cheating here a little bit because I've talked about quantitative ICP and I've not really told you about our relative changes that is based on transfer function analysis, but the two are very much related. But to give you an idea of just very preliminary hospital data, we measure changes in intracranial pressure, which we downsample typically. We measure oxy and deoxyhemoglobin, and oxyhemoglobin is very much related to cerebral blood flow that I just described. And while we are not quantitative, we're one step behind, but in the hospital data, what we have found is that even oxyhemoglobin can be used to at least look at relative changes in intracranial pressure based on transfer function analysis that I haven't described here. Really the purpose of this is saying that we are in a hospital, we're analyzing the DCS data right now to come up with the quantitative description as well for humans, not just the animals. So really briefly, I do want to talk about the second part of this talk, which is how is any of this related to neuronal function? Why do we care about intracranial pressure and cerebral autoregulation in terms of then understanding and optimizing neuronal function in humans? And so really the question we asked, and that work is mostly done by a graduate student, Deepshika, who put this really nice slide together. I think is very intuitive, saying that we ask does impaired autoregulation impact neurovascular coupling? And so ultimately, the way we answer this is by saying we measure neuronal function. That's done with electroencephalography. We measure the vascular response. That's near-infrared spectroscopy, so oxy and deoxyhemoglobin. And we're measuring changes in autoregulation based on blood flow. So that's done with diffuse correlation spectroscopy. So in the same animal model that I just talked about, we also perform, so we have the intracranial pressure sensor, we have the cannula to change intracranial pressure by putting fluid inside the head, and we alter, we go up and down in intracranial pressure, which leads to changes in cerebral perfusion pressure. And in addition to DCS measurements, we add NIRS and EEG, so that's individual cortex, to then measure visual evoked responses in those animals. So the way we do this is by means of checkerboards of visual stimuli. Specifically, as I said before, what we want to know is how does neurovascular coupling, how is neurovascular coupling related to cerebral autoregulation? So the paradigm is we have a rotating checkerboard, which we flash for about 5 seconds. And then we give the animals about a 10-second break and repeat it multiple times. And we do that at every perfusion pressure level. So ultimately, what we want to know is how does the neuronal function and the vascular or neuronal task-related visual evoked response, how does that look like, as well as how does the vascular response look like as a function of perfusion pressure. So what happens when perfusion pressure is such that autoregulation is maintained versus not maintained? What happens to neuronal function? And to show you that overall we can actually measure this impairment of autoregulation, this is a group average over multiple animals, where blood flow is maintained between about 60 to 110 millimeter mercury. And outside that range, blood flow is no longer maintained. And we have hyperperfusion of the brain. So then for each level of perfusion pressure, what you're going to be seeing is the neuronal response. So that's the visual evoked potential to the flickering checkerboard. So it's essentially a flash of this checkerboard that produces a neuronal activation, as well as then the vascular response as measured in this case with total hemoglobin. So that's oxy plus deoxy combined as a function of time. So what we found is that when perfusion pressure is relatively low, so that's somewhere in the autoregulatory range, the visually evoked responses look more or less the same. And at really high perfusion pressure, so outside the autoregulatory range, the visual evoked potential does look visually just very, very distinctively different from the rest of them. In addition, what we found is that the vascular response, again, that's the vascular response to neuronal function, is also altered depending on how high perfusion pressure is. So really what the slide says is we see that the brain is responding differently depending on which pressure environment it is in. But what I'm showing you here is not asking them specifically what is the coupling between neurons and the vasculature. And so in order to address this, what we really did was we said, okay, let's take the EEG signals. In particular, we're taking the amplitude, the EEG amplitude squared from the EEG amplitude. We take the hemodynamic signals, so that's what is called a block average. And then we do convolution with the hemodynamic response function, or we're fitting the hemodynamic response function that best describes the coupling between EEG and NIRS, or neuronal and vascular responses. So, which is then evaluated on the hemodynamic response. So that's then the dotted line. The dotted line is the convolution between EEG and the fitted hemodynamic response function. So what we did was then saying, well, what is the response function? So it's ultimately an impulse response function. How does the response function, the hemodynamic response function, look like as a function of perfusion pressure? So as a function of autoregulation. So within about 60 to 100 millimeter mercury, the shapes look all very much the same. Outside of that range, we start seeing changes in the coupling between, so that's the changes in the hemodynamic response function, therefore changes in the coupling between neurons and the vasculature. And so the green here is really when autoregulation is intact versus red is impaired. So a different way of showing this is on top of this autoregulatory curve, which has a name, by the way, it's called the Lawson's curve. But essentially saying that whenever blood flow, cerebral blood flow is maintained, so that corresponds to a particular perfusion pressure, then the coupling between neurons and the vasculature is maintained. Outside that range, the coupling is altered, is different, and we can quantify that with the hemodynamic response function. Now, clinically, this is important because I've just talked about a lot of different concepts and a lot to be understood here in terms of from a clinical perspective, but really taking a step back, what that implies is I can take an EEG sensor, so those are just a few electrodes, take an optic sensor to measure the hemodynamic signals. All of them are bedside compatible. And the coupling between the EEG and the NIRS, or the neuronal and the hemodynamic signals, goes hand in hand with perfusion pressure and autoregulation. So therefore, we can build based on this coupling, we can come up with a biomarker that could be potentially used to actually optimize treatment. So that could be lower intracranial pressure, or change blood pressure medication, or possibly predict which patient might have to go into the surgical room for craniotomies to reduce pressure. So in conclusion, what I've told you is we came up with a non-invasive alternative to measure intracranial pressure that's based on a cardiac waveform of cerebral blood flow. And I have shown that neurovascular coupling is linked to cerebral perfusion pressure and could possibly be used as a biomarker for cerebral health, going hand in hand with perfusion pressure and autoregulation. So with that, I would like to thank my absolutely amazing, awesome lab, very talented graduate students and postdocs, and a bunch of collaborators, in particular for this project, the hospital collaborators and funding sources. And I'd be happy to take questions.
Jana, thank you. This was really nice. I'll clap for everybody. And I think now and I, we can take turns in pitching questions to you. And actually what we should be doing is a couple of different things. You can put your questions in the Q&A. You can use the chat. We'll look at that. But you can also just raise your hand and we can give you speaking rights, which actually should be able to allow you to unmute yourself, I think. So keep the questions coming. It's a lot of good mechanical engineering thinking that you can now try out on Jana. Actually looking at a different, oh, there's a question here. So we have one from Shurik, I think.
Hello. Where do you see questions? Or is it just raising hands?
There's a Q&A.
I have a couple small questions. Also, thank you and hi.
Hi.
But for the hospital probe with the kids, there was looked like the DCS separations were much smaller than NIRS.
Hmm.
What were the differences in separations?
The reality is, like a typical PI, I only have old photos.
Okay, yeah.
So realistically, that was an old probe. Now the combination, since you know specifically what I'm talking about, but we're using a multi-distance nearest probe that goes from 1.5 to 3, I believe, centimeters, and then the DCS is about 2.5 centimeters.
And then the other question was, this from like early in the talk, was you showed when the intracranial pressure was changed and like oscillated, you know, both the NIRS and the DCS were oscillating as well, and it seemed like I think I just didn't have time to read the axes, but it seemed like the NIRS had a stronger response to change in ICP, and I was wondering like which one of the two kind of has a stronger, because they both oscillated, it seemed, with the ICP changes.
They both do, and that again was an old figure where the signals from the blood flow for the DCS were still measured, if I remember correctly, the single mode fibers. Ever since we switched to few mode fibers, so the signals did become much stronger. The relative contribution, so oxyhemoglobin and blood flow are very much related in terms of which of the two has the best, better SNR. I would have to go back. I do believe that CBF actually ended up having the better SNR to oscillations in ICP.
Okay, because that's what I thought like would make sense, but it was. But anyways, thank you. Those are my.
But the details are all in this case in Alex's JCBFM paper from 2019. So we have SNR curves for that if you're interested.
Thank you.
Jana, for the diffuse correlation spectroscopy, you go through quite a bit of tissue, right? You have the source, and then you have the detector. So you're averaging over different blood flow in different sort of vasculature sizes and like this, or?
Yeah. No, it's definitely a lump sum of arterioles, capillaries, and venules, and it's definitely the decay of that autocorrelation function is a weighted average of all of them combined. We're not sensitive to large vessels because blood flow would be too fast and the decay is too fast, and the detectors wouldn't actually be able to measure those decays. But overall, with NIRS and with DCS, with both instruments, it's always a combination of all three compartments, and we don't really have a good way of differentiating. Yeah.
But the changes that you're seeing are going to be more substantial between the different pressure regimes than the variations within these different types of vasculature. And I guess that's the answer is probably yes to that because it works. But
yeah,
I wanted to ask something else, and that is about.
Sorry, one more thing about this is you're right. Different pressures will influence the venules versus arterioles differently because of them being pressurized. For the ICP extraction itself, we're looking at the cardiac waveform, and the cardiac waveform only ever happens in the arterioles. So that signal comes from the arterioles because venules don't pulsate. But that's specific for the cardiac pulsation. But sorry, what was your other question?
No, I think there's Anthony is actually going up. He had a question, and I should leave him going first. But I have one more that I want to ask.
He did not have mic access. He did not get mic access.
He should have it now. Let's see.
I think he still muted himself. Anthony, there should be a little pop-up asking you to unmute, or we can read your question as you prefer. Okay, shall we read his question then?
Oh, he doesn't have a mic. So what he's saying is, and this is Anthony, I'm paraphrasing him. The question is a bit. I'm curious if you see any hysteresis in the neuronal response. Does it matter if you are increasing, decreasing, or making large jumps in pressure? So how does this respond to different pressure inputs?
That is a brilliant question. A complicated one to address. First of all, so we actually, little detail here, we change intracranial pressure, but anything I've shown you is in a correlation with perfusion pressure. So that ultimately means really the pressure difference. What happens oftentimes is that when we go up with intracranial pressure, slowly but surely blood pressure will also increase, which then has an effect on this pressure differential. So ICP is not really the driving factor. We do not see fast changes in neuronal function with fast intracranial pressure changes. So in that sense, we're limited to potentially also slow drifts in CPP. And it's a very vague answer to ultimately be saying, awesome question, I don't have an answer, based on how we set up the experiment and how limited we are in terms of time chunks of stable CPP. Because, like, as you can imagine, physiologically, a lot of things are changing all the time. Yeah.
Jana, a question on the NIRS. That system is pretty amazing. It looks like you can buy this somewhere, but you mentioned it's one of the students who built Charles, I guess.
Yeah.
So that's a hidden compliment for how cool that came out. Why do you need 4 different collection points? What do you gain by doing that?
It's an SNR question. So for the 2 different instruments, the NIRS, the commercial one, uses 4 different source detected distances. For this particular instrument, which is called the frequency domain NIRS system, so it's ultimately based on diffusion of photons. If you send that's the bananas. Nevin, I didn't even show bananas.
No bananas.
I know the probability density function of how deep photons go depends on the source detector distance. So the further the distance, the deeper the photons. If you use 4 detectors combined, plus some other information, you can actually quantify the optical properties, and that's why we need 4 distances, but that's for a separate part. The DCS part with the blood flow system, the reality is the detectors are single photon counting modules. We are working with very low light levels, because we need to use single or few mode fibers. So ultimately we just average 4 channels together to get one good SNR channel. So it's really just a question of SNR. But for shorter distances, if you don't need to go into the brain, let's say, which is about 15 millimeters from the surface, you can get away with single channels, and you don't need more than one.
Cool.
I'm curious, Jana, the translatability of this research is really obvious to the clinical use. How small could this system potentially go? Does it have to be this large bundle of cables descending off a skull cap on the patient, or is there any hope to get it to be as small as a pulse oximeter is today? And I don't mean that facetiously, but reasonably packaged.
So there are multiple avenues. The ICP extraction, or also like some of the work that Jason is doing on impedance measurements on the microvasculature, that's all based on blood flow, which is the diffuse correlation spectroscopy. And there are a few buzzwords that I threw out, which is long coherence laser, as well as single photon counting modules. Those are pretty expensive and also pretty large footprint. And so the long coherence lasers, I am not aware of anyone really building them in any kind of small form factor. So they're about like that big. And then the modules, so it's like Jason's design right now is about a shoebox with then the fibers coming out. I'm sure that this can be built smaller. It's just a question of time, but not with the current available components. The nearest part, yes, there are really cool companies also nowadays that make very, very nicely integrated helmets and such, but so that can definitely be much smaller.
Oh, thank you.
Yeah, no, thank you. We're running a bit over. It's 5:03 now. So I think that would be a good time to officially say thank you for a great seminar and for speaking to us today. Of course, if anybody has questions, we will stay on for a little while and we can promote you to speak and talk so we can ask Jana more questions. Thanks for joining us today.
Yeah, thank you. Thanks for having me.